Chapter 1 Cell, the unit of life. Point 1 Cell is the fundamental structural and functional unit of all living organism. Point 2 Anton von Leeuwenhoek first saw and described a living cell, and Robert Brown discovered the nucleus. 3 Skladin and Swan together formulated the cell theory. Point 4 Omnis cellula acellulo was explained by Rudolf Virchow. 5 Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that have membrane-bound nuclei are called eukaryotic, whereas cells that have lack a nuclei are called prokaryotic. Point 6 Ribosomes are non-membrane-bound cell organelles found in all cells. Point 7 The smallest cell in mycoplasma of 0.3 micrometer in length. Point 8 The four basic shapes of bacteria are called bacillus, rod-like, coccus, spherical, vibrio, comma-shaped and spirillum को स्पैरा बोलते हैं. 9 A typical eukaryotic cell is of 10 to 20 micrometer and the size of viruses is 0.02 to 0.2 micrometer. Point 10 Many bacteria have a small circular DNA in addition to genomic DNA and is called plasmid. Point 11 In bacteria, the infoldings of cell membrane is called mesosome, which of the characteristics of prokaryotic. 12 The cell envelope of prokaryotic cell consists of 3 layers of glycolysis, cell wall and plasma membrane. Point 13 In some prokaryotics, glycolysis could be loose sheet called slime layer, and sometimes it may be thick and tough called capsule. Point 14 In bacteria, cell wall determines the shape of cell, provides structural support. Point 15 In cyanobacteria, the membranous extension into cytoplasm called chromatophores contain pigment. Point 16 Motile bacteria have thin filamentous extension from the cell wall called flagella, which is composed of 3 parts: filament, hook, basal body, in which filament is the longest portion and extend from cell surface to the outside. 17 Surface structure present in bacteria and flagella, pili and fimbria. 21 Ribosomes are about 15 nanometer by 20 nanometer in size and are made up of 2 subunits. Prokaryotes have 70 S ribosomes. 15 ribosomes are about 15 nanometer by 20 nanometer in size and are made up of 2 subunits. Prokaryotes have 70 S ribosomes. 70 S ribosomes 50 S plus 30 S सबयूनिट से मिलकर बनता है. Various eukaryotes have 80 S ribosomes. 18 ribosomes are about 15 nanometer by 20 nanometer in size and are made up of 2 subunits. Prokaryotes have 70 S ribosomes. 50 S plus 30 S सबयूनिट से मिलकर बनते हैं. Various eukaryotes have 80 S ribosomes. 60 plus 40 S सबयूनिट से मिलकर बनते हैं. 9 Several ribosomes may attach to single mRNA and form a chain called polyribosomes and polysomes called. 20 Reserved material in prokaryotic cells stored in cytoplasm in the form of inclusions body. When the cell membrane is composed of lipids that are arranged in a bilayer, the membrane also contains protein and carbohydrates. 22 In humans, the membrane of the erythrocytes has approximately 52% protein and 40% lipid. 23 According to fluid mosaic model, the quasi-fluid nature of lipids enable the lateral movement of proteins within the overall bilayer. This model was given by Singal and Nicholson in 1972. 24 Movement of water by diffusion across plasma membrane is called osmosis. 5 Sodium potassium pump is an example of active transport. 6 The primary cell wall of a plant cell is capable of growth and secondary cell wall is formed on inner towards membrane. 26. The primary cell wall of a plant cell is capable of growth, and the secondary cell wall is formed of inner का मतलब towards membrane side of the cell. 27. The middle lamella is a layer chiefly made up of calcium pactate which glues neighboring cells together, and plasmodesmata connect the cytoplasm of neighboring cells. Point 26. The primary cell wall of a plant cell is capable of growth, and secondary cell wall is formed of inner towards membrane side of the cell. Point 27. The middle lamella is a layer chiefly made up of calcium pactate which glues neighboring cell together, and plasmodesmata connect the cytoplasm of neighboring cells. Point 28. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. 29. The ER-bearing ribosomes on their surface is called RER. 30. The smooth endoplasmic reticulum is the major site of synthesis of. Point 31. The Golgi cisternae are concentrically arranged near the nucleus with distinct convexes or the forming phase and concave trans or maturing phase. 32. The vesicles from the ER fuse with this phase of Golgi apparatus and move towards the maturing trans phase. 33. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. 34. The lysosomal vesicles are rich in hydrolytic enzymes that are optimally active at acidic pH. 35. The vacuole is bounded by a single membrane called tonoplasm. Vacuole is important for excretion, and in protists, food vacuoles are formed by engulfing the food particles. Chloroplasts are double membrane bound structures. Mitochondria is called matrix and possess a single circular DNA molecule. 9. The cell organelle mitochondria is the site of aerobic respiration, and it divides by fission based on the pigments into chloroplast, chromoplast, and leucoplast. Types of leucoplast store starch, carbohydrate, elioplast. Last type of leucoplast store starch, carbohydrate, elioplast store oils and fats. Various eleuroplast store proteins. Pseudomonas, a green algae, has one chloroplast per cell. By inner membrane of the chloroplast is called stroma. Arranged in stacks like the piles of coins called grana, and are connected to other thylakoids of different stacks by membranous tubules called stroma lamellae. It is the site of light reaction, and the stroma is of dark reaction. The ribosome of chloroplast and mitochondria are of 70S type. Ribosomes are composed of ribonucleic acid and proteins. Ribosomes are 80S types. Here S is eukaryotic ribosomes are 80S type. Here S is Wedberg's unit, which stands for the sedimentation coefficient. Eukaryotic flagella is called axonium, which consists of 9 pairs of doublets of radially arranged peripheral macrotubules and 1 pair of centrally located macrotubules. The arrangement of macrotubules in eukaryotic flagella is referred to as 9+2. Central tubules are connected by bridges and is enclosed by central sheath, which is connected to one of each peripheral doublets by radial spokes. Cilium and flagella emerge from central-like structure called the basal, made up of nine evenly spaced peripheral plate fibrils of tubulin protein. Proximal region of the centriole is called the hub, which is connected with the tubules of peripheral triplets by radial spoke. Between two parallel membranes of nuclear envelope is called perinuclear space. Their matrix is called nucleoplasm, which contain nucleolus and chromatin. Nucleolus is the site of ribosomal RNA synthesis. The interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatins, which contains DNA and some basic proteins called histones. Position of centromere chromosomes Position of centromere chromosomes are metacentric, subcentric, acrocentric, and telocentric. Primary construction of chromosome is called centromere. Sometimes a few chromosomes have secondary construction which gives the appearance of a small fragment called the satellite. ये वीडियो इंग्लिश में है एंड अगर आप हिंदी में प्रेफर समझना चाहते हो तो एक हिंदी वाला लेक्चर भी है, आप वो भी देख सकते हो।